Hello everyone, my name is Shamus Corrigan, and for my independent research project with the Bioscience Technology program at Holland College, I investigated the effects of raspberry polyphenols on the polarization of THP1 macrophages. So polyphenols are compounds found in plants that have antioxidant, anti-inflammatory, and anti-cancer properties. One plant with a unique phenolic makeup is Rubus idaeus, also known as the red raspberry. There are many unique anthocyanins and elegotannins present in raspberries, and they also have a history of being used in medical applications dating all the way back to the Roman era. And so looking into these medical applications and the immune system, we have the macrophage. The macrophage is a type of white blood cell which has many roles, including phagocytizing foreign microbes, releasing signaling molecules, and promoting tissue repair. There are two main states of the macrophage. The M1 and M2 states both are derived from the M0 state. The M1 state is associated with pro-inflammatory and a destructive immune response, while the M2 state is associated with tissue repair and an anti-inflammatory immune response. Two prominent cytokines associated with macrophages are TNF-alpha and IL-10 for M1 and M2 states respectively. And the ability to switch between these two states is extremely important for the body, as an overexpression of either can be very dangerous. An overexpression of the M1 state can cause diseases such as rheumatoid arthritis, while an overexpression of the M2 state can promote tumor growth and proliferation. Therefore, I was interested in the effect that the unique raspberry phenolic profile could have on the polarization of the macrophages. It was hypothesized that after treatment with raspberry phenols, it would inhibit the polarization of M0 macrophages towards the M1 state. This was accomplished by first extracting the raspberry polyphenols. Using a solvent extraction method followed by SBE for separation, and then DPPH and FNC assays were performed for phenolic quantification and antioxidant activity. THP1 cells were then differentiated into M0 macrophages with PMA. Respective cells were treated with the phenolic extract, and then respective cells were polarized with LPS. A TNF-alpha ELISA was performed to detect TNF-alpha levels, and PCR was performed to observe TNF-alpha and IL-10 gene expression rates. All the data was then analyzed using a two-way ANOVA. So the main result of the study found that after treatment there was a significant decrease in TNF-alpha expression and production in the macrophages. The ELISA found a significantly decreased concentration of TNF-alpha in the media being tested, while PCR found significantly decreased TNF-alpha gene expression rates in the cells. Of course, both of these results corroborating each other. And then the results from DPPH and FNC assay also indicated phenolic compounds present and antioxidant activity within the samples. Finally, the relative IL-10 gene expression was found to have no significant change between the treated and non-treated cells. So overall, it was found that raspberry phenolic treatment was able to decrease expression and production of TNF-alpha, indicating a decrease in the M1 polarization after treatment, though no significant change was found in the IL-10 gene expression, indicating that treatment was not able to polarize towards an M2 state, only to prevent the M1 state polarization, supporting the hypothesis. So some future research that could be conducted similar to this would be to look at the specific compounds found in the phenolic makeup being tested, and perhaps separate them further to see which have the greatest effect in replicating the results obtained here, or performing the experiment using similar fruits with unique phenolic compounds in themselves. And finally, an interesting, if not more complicated idea would be to create a disease model, perhaps replicating rheumatoid arthritis, and see what effect treatment may have on that. And so finally, I want to thank Jesse Livingston, Mike Gibson, Crystal McFadgen, Marla McKenzie, Jennifer Slemmer, and Shonda Solis Chavez for guiding and supporting me throughout the Bioscience Technology program. Thank you.